Hello everyone and welcome to this explainer. I am absolutely thrilled you're joining us today because we're going to unpack a major, real game changer in the world of genomics. Now, if you're actively working with genetic mapping, population evolution, or really any complex genome project, you already know that reduced representation whole genome sequencing is an absolutely critical tool. But let's be totally honest here. It hasn't always been the smoothest ride. So today, we're diving deep into a revolutionary approach known as 2b-RAD sequencing. This is a technique that is fundamentally rewiring how we tackle the persistent challenges of high-throughput SNP genotyping.
Imagine a workflow where you could completely bypass the usual headaches. What if you could just entirely eliminate data redundancy and those incredibly frustrating non-uniform fragments in your genotyping workflows? Anyone who has spent days or, let's be real, sometimes weeks, wrestling with traditional genetic mapping data knows exactly what a massive drain that is. It eats up your resources, it wildly complicates your bioinformatics, and ultimately it just delays your results. But what if we could just bypass all of that?
All right, so here is our roadmap for today. First, we'll look at the genotyping bottleneck. Then we'll enter the world of 2b-RAD technology and break down its workflow. After that, we'll see exactly why 2b-RAD wins out over older methods, explore the specialized 2b-RAD services from CD Genomics, and finally, talk about how to accelerate your next big project.
Okay, let's kick things off with part 1, the genotyping bottleneck.
So traditional RAD-Seq processes are, well, they're inherently a bit messy. They rely heavily on physical interruption methods like sonication, along with arbitrary fragment size selection, which usually sits somewhere between 300 to 1,000 base pairs. Plus, you have these incredibly complex, multi-step DNA isolation and purification processes. And the result of all that, you get highly divergent fragment lengths and completely non-uniform sequencing depths across your different tags. To make matters worse, the data analysis quickly turns into a nightmare of false positives caused by repetitive sequences, and those older methods really struggle to filter them out effectively.
Moving right along to Section 2, enter 2b-RAD technology.
The crucial starting point here is understanding exactly what 2b-RAD actually is. Originally described back in 2012, the 2b-RAD or IIb-RAD method utilizes type IIB restriction endonucleases. Enzymes like BsaXI or BsaXI are literally the stars of the show here. And what makes them so remarkable? Well, they recognize specific sites on double-stranded DNA, and then they brilliantly cleave the genomic DNA on both sides of those recognition sites. This generates fixed-size, double-stranded DNA fragments with protruding, non-cohesive ends. It's just a beautifully precise molecular mechanism.
And right away, you see the immediate payoff. Because the enzyme cuts at fixed distances on both sides, it generates incredibly uniform fragments ranging strictly from 33 to 36 base pairs. Think about that for a second. This means you completely eliminate the need for physical interruption and those finicky size selection steps. Now, even though you might generate fewer fragments compared to single enzyme RAD approaches, the sheer consistency of these short tags gives you superior resolution, massive coverage, and totally uniform amplification efficiency during PCR.
Which brings us to Section 3, the 2b-RAD workflow.
The library construction for 2b-RAD is wonderfully streamlined into 4 major stages. First up, we have the digestion step, which often uses that BsaXI enzyme we mentioned. Once those uniform fragments are created, we move right into ligation, where specific adapters are attached to those protruding cohesive ends. Step 3 is amplification, utilizing high-fidelity DNA polymerase and a set of primers. And finally, barcoding. These primers actually introduce sample-specific barcodes during PCR, which means we can pool massive amounts of samples together super efficiently for the sequencer.
Looking at the chronological flow, you kick off with genomic DNA extraction, obviously checking for concentration and making sure there's no degradation. Then, you move through digestion and adapter connecting. After amplification, the qualified libraries are sequenced, frequently using Illumina paired-end 150 base pair technology. But the workflow doesn't stop at the sequencer. Quality control is absolutely vital here. The system actively excludes terminal tag positions to eliminate artifacts, and it completely scrubs out long homopolymer regions and low-quality reads. So what you're left with are purely high-quality reads, fully ready for precise alignment.
All right, let's dive into Section 4, Why 2b-RAD Wins.
When you stack these technologies head-to-head, the contrast is honestly striking. With traditional RAD-Seq and 2b-RAD, fragment sizes are just naturally non-uniform, which leads to wildly divergent tag sequencing depths. It's incredibly uneven. But with 2b-RAD, both the fragment size and the tag sequencing depth are wonderfully uniform. And when it comes to data analysis, while older methods get bogged down by false positives from repeated sequences, 2b-RAD brings in a new data typing algorithm called IML. This effectively slices right through the noise, completely removing the distraction of those false positives. The list of technical benefits just keeps stacking up. For instance, because 2b-RAD tags are independent, since the contact point is on both sides of the restriction sites, two tags literally can't be completely adjacent. This drastically reduces data redundancy. Also, the entire library is sequenced, so absolutely no restriction sites are lost. It's remarkably robust too, allowing for low DNA input and even degraded DNA. And the density is just phenomenal. In a 1G genome, you can generate over 200,000 tags with an average distribution distance of just 2 kilobases. Plus, it effectively utilizes both SNPs and dominant markers.
And I really want to highlight this specific number, 95%. 2b-RAD employs an N+1 strategy for technical repeats. What that actually means is the same sample repeats the library sequencing construction to absolutely guarantee that the tag recurrence rate of that same sample is no less than 95% between two experiments. That kind of technical repeatability is a massive promise, and frankly, it's one you simply can't ignore in rigorous research.
This seamlessly brings us to Section 5, CD Genomics 2b-RAD Services. Now, understanding this incredible methodology is one thing, but actually executing it perfectly in-house, that can be intensely resource draining. You need highly specialized personnel, and the bioinformatics bottlenecks alone can literally stall a project for months. And this is exactly where a partner like CD Genomics changes the whole equation. They offer a complete end-to-end service, from the very first genomic DNA quality check all the way to complex population diversity assessments. They manage the entire pipeline with incredibly strict quality management. You get confident, unbiased results without the internal struggle.
Let's look at their specific, guaranteed sequencing strategy specs. When you partner with them, they're utilizing top-tier Illumina platforms, like the high-Seq X10 for PE150 sequencing. They target a genome label average density of 2 kilobases, and on top of that, they guarantee an average label depth of 20x or greater, boasting a classification accuracy of 95% or higher. Basically, this is high-density, high-precision genotyping operating reliably at a very impressive scale.
But their bioinformatics pipeline is where the magic really happens for researchers. They don't just dump raw data on your desk and wish you luck. Their expert team takes you from raw data QC right through sophisticated SNP genotyping and locus annotation. From there, they can construct high-precision genetic linkage maps, execute quantitative trait locus or QTL fine mapping, and even conduct comprehensive population evolution analyses based on allele frequencies. They handle the heavy lifting.
Finally, let's wrap up with Section 6, Accelerate Your Next Project.
As they stayed right in their own materials, CD Genomics provides a comprehensive service that encompasses the entire workflow from genomic DNA quality control to the determination of genotypes and the assessment of population diversity. And actually, it's extremely important to note here that they are a fully licensed provider of this advanced technology. That ensures full legal compliance with the sequence-based genotyping patents held by Keygene NV. So you're getting scientific excellence that is also completely legally safeguarded. Which leaves us with one final, vital question to ponder today. Are you ready to finally eliminate data redundancy and unlock reliable, high-density SNP genotyping for your next complex genome project? If you're tired of banging your head against the bottlenecks of traditional RAD-Seq and want the uniform precision of 2b-RAD, it's absolutely time to leverage this workflow. I'd highly recommend reaching out to the experts at CD Genomics with your customized requests to see exactly how they can accelerate your research. Thank you so much for joining me for this explainer. And hey, happy sequencing!